हेलो फ्रेंड्स, मैं अनुराग कुशवाहा और मैं पॉडकास्ट बना रहा हूँ आप लोगों के लिए। और चैप्टर है एक्सक्रीटरी प्रोडक्ट्स एंड देयर एलिमिनेशन। आज मैं ना सिर्फ इस चैप्टर को रीड करके आपको सुनाऊंगा, बल्कि साथ ही में इस चैप्टर को अच्छी तरह से एक्सप्लेन भी करूँगा। और जहाँ कहीं भी आप हों वहाँ से आप इसे शांतिपूर्वक आँख बंद करके भी सुन सकते हैं, या आप चाहें तो आप टहलते हुए भी सुन सकते हैं। कहीं भी आप बस ईयरफोन लगाइए और मेरी बातों को सुनते रहिए। तो अब करते हैं स्टार्ट। Animals accumulate ammonia, urea, uric acid, carbon dioxide, water and ions like Na+, K+, Cl-, phosphate, sulfate, etc. either by metabolic activities or by other means like excess ingestion. These substances have to be removed totally or partially. In this chapter, you will learn the mechanism of elimination of these substances with their special emphasis on common nitrogenous waste. Ammonia, urea, and uric acid are major forms of nitrogenous waste. Excretory products by animals. Ammonia is the most toxic and require large amount of water for elimination, whereas uric acid, being the least toxic, can be removed with minimum loss of water. Process of excreting ammonia is ammonotelism. क्या process of excreting ammonia is ammonotelism. Many bony fishes, aquatic amphibians and aquatic insects are ammonotelic in nature. क्या many bony fishes, aquatic amphibians and aquatic insects are ammonotelic होते हैं। अमोनोटेलिक मतलब जिसमें अमोनिया, जिसका एक्सक्रीटरी प्रोडक्ट्स अमोनिया होगा। Ammonia is a readily soluble, is generally excreted by diffusion across body surface or through gill surface. क्या ammonia is, as it is readily soluble, is generally excreted by diffusion across body surface or through gill surface. In fish as ammonium ions, kidney do not play any significant role in its removal. क्या किडनी डू नॉट प्ले एनी सिग्निफिकेंट रोल इन इट्स रिमूवल, मतलब किडनी कोई भी सिग्निफिकेंट रोल प्ले नहीं करता है अमोनिया को रिमूव करने में। टेरेस्ट्रियल एडेप्टेशन नेसेसिटेटेड द प्रोडक्शन ऑफ लेसर टॉक्सिक नाइट्रोजनस वेस्ट लाइक यूरिया, मतलब जब हम टेरेस्ट्रियल की तरफ एडाप्ट हुए, मतलब हम या कोई भी लिविंग्स, तो वो उसका जो नाइट्रोजनस वेस्ट है वो कम टॉक्सिक हुआ, जैसे कि यूरिया और यूरिक एसिड्स। एंड now mammals, many terrestrial amphibians and marine fishes mainly excrete urea and are called ureotelic animals. Ammonia produced by metabolism is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out by the kidneys. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolarity. Reptiles, birds, land snails, and insects excrete nitrogenous waste as uric acid in the form of pellet or paste with the minimum loss of water and are called uricotelic animals. A survey of animal kingdom present a variety of excretory structure in most of the invertebrates. These structures are simple tubular form, whereas vertebrates have complex tubular organs called kidney. Some of these structures are mentioned here. Protonephridia or flame cells are the excretory structure in platyhelminthes. क्या Protonephridia or flame cells are excretory structure in platyhelminthes. And the bracket में flatworms, example planaria rotifus, some annelids and the cephalochordates amphioxus. Protonephridia are primarily concerned with ionic and fluid volume. Protonephridia are primarily concerned with ionic and fluid volume regulation, osmoregulation. Nephridia are the tubular excretory structure. Nephridia are the tubular excretory structure of earthworm and other annelids. Nephridia help to remove nitrogenous waste. नेफ्रेडिया हेल्प करता है रिमूव करने में नाइट्रोजनस वेस्ट को एंड मेंटेन अ फ्लूइड एंड आयनिक वैलेंस. Malpighian tubules are the excretory structure of most of the insects including cockroach. Malpighian tubules help in the removal of nitrogenous waste and osmoregulation. Antennal glands or green glands perform the excretory function in crustacean like prawns. और अब हम लोगों का नया पॉइंट है ह्यूमन एक्सक्रीटरी सिस्टम. In human, the excretory system consists of a pair of kidney, one pair of ureter, a urinary bladder, and a urethra. Kidney are reddish brown, bean-shaped structure situated between the level of last thoracic and third lumbar vertebra, close to the dorsal inner wall of abdominal cavity. Each kidney of an adult human measures 10 to 12 cm in length, 5 to 7 cm in width, and 2 to 3 cm in thickness, with an average weight of 120 to 170 gram. Toward the center of the inner concave surface of the kidney is a notch called hilum, through which ureter called vessels and nerves enter. Inner to the hilum is a broad funnel-shaped space called the renal pelvis with projection called calyces. The outer layer of kidney is a tough capsule. Inside the kidney there are two zones, an outer cortex and an inner medulla. The medulla is divided into a few conical masses, medullary pyramids, projecting into the calyces. The cortex extends in between the medullary pyramids as the renal column called column of Bertini. Each kidney has nearly 1 million complex tubular structure called nephrons. क्या each kidney has nearly 1 million complex tubular structure called nephrons, which are functionally units. नेफ्रॉन क्या होता है फंक्शनल यूनिट होता है किडनी का. इच नेफ्रॉन हैज़ टू पार्ट्स. हर नेफ्रॉन के पास दो पार्ट होता है. एक होता है ग्लोमेरुलस और एक होता है रेनल ट्यूब्यूल्स. ग्लोमेरुलस इज टफ्ट कैपिलरी. ग्लोमेरुलस क्या होता है टफ कैपिलरी का फॉर्म होता है बाय द एफरेंट आर्ट्रियोल अ फाइन ब्रांच afferent arteriole का a fine branch of renal artery. Blood from the glomerulus is carried away by an efferent arteriole. The renal tubule begin with a double wall cup-like structure called Bowman's capsule. Renal tubules begin with a double wall cup-like structure called Bowman's capsule. Which includes the glomerulus. Glomerulus along with Bowman's capsule is called the Malpighian body or renal corpuscle. The tubule continues further to form a highly coiled network proximal convoluted tubule. तारू बदनाम करती। अब हम पढ़ेंगे अब a hairpin shaped Henle's loop is the next part of the tubule which has a descending and an ascending limb. The ascending limb continues as another highly coiled tubular region called distal convoluted tubule DCT. The DCT of many nephrons open into a straight tube called collecting duct, many of which converge and open into the renal pelvis through medullary pyramids in the calyces. The Malpighian capsule, PCT and DCT of the nephron are situated in the cortical nephron of the kidney, whereas the loop of Henle dips into the medulla. In majority of nephrons, loop of Henle is too short and extend only very little into the medulla. क्या इन मेजॉरिटी ऑफ नेफ्रॉन क्या मेजॉरिटी ऑफ नेफ्रॉन ज़्यादातर नेफ्रॉन्स में लूप ऑफ हेनले इज टू शॉर्ट बहुत छोटा होता है एंड एक्सटेंड ओनली वेरी लिटिल इनटू द मेडुला और बहुत छोटा होता है मेडुला में। सच नेफ्रॉन आर कॉल्ड कॉर्टिकल नेफ्रॉन और ऐसे नेफ्रॉन को कॉर्टिकल नेफ्रॉन कहते हैं। इन सम नेफ्रॉन और कुछ नेफ्रॉन में लूप ऑफ हेनले बहुत बड़ा होता है जो रन करता है डिप इन द मेडुला के अंदर भी चला जाता है और ऐसे नेफ्रॉन को जस्ट मेडुलरी नेफ्रॉन कहते हैं। द इफरेंट आर्ट्रियोल इमर्जिंग फ्रॉम द ग्लोमेरुलस फॉर्म इफरेंट आर्ट्रियोल इमर्जिंग फ्रॉम द ग्लोमेरुलस फॉर्म a fine capillary network around the renal tubule called peritubular capillary. A minute vessel of this network runs parallel to the Henle's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in cortical nephron. यूरिन फॉर्मेशन अब हम पढ़ेंगे अबाउट नाउ वी आर टॉकिंग अबाउट यूरिन फॉर्मेशन इन्वॉल्व्स three main process namely glomerular filtration, reabsorption and secretion. क्या glomerular filtration, reabsorption and secretion that takes place in different part of the nephrons. The first step in urine formation is the filtration of blood. First step in urine formation is the filtration of blood, which is carried out by glomerulus and is called glomerular filtration. On an average, 1100 to 1200 ml of blood is filtered by the kidney per minute, which constitutes roughly 1/5 of the blood pumped out by each ventricles of the heart in a minute. Glomerular capillary blood pressure causes filtration of blood through three layers: endothelium of glomerular blood vessels, the epithelium of Bowman's capsule, and a basement membrane between these two layers. The epithelial cells of Bowman's capsule, called podocytes, are arranged in an intricate manner so as to leave some minute spaces called filtration slits or slit pore. Blood is filtered so finely through these membranes that almost all the constituent of the plasma except the proteins pass on to the lumen of the Bowman's capsule. Therefore, it is considered as a process of ultrafiltration. Amount of the filtrate formed by the kidney per minute is called glomerular filtration rate GFR. GFR in a healthy individual is approximately 125 ml per minute. GFR in a healthy individual is approximately 125 ml per minute and 180 liter per day. How many? 180 liter per day. The kidney have built in mechanism for the regulation of glomerular filtration rate. Kidney have built-in mechanism of for the regulation of glomerular filtration rate. One such efficient mechanism is carried out by juxtaglomerular apparatus JGA. JGA is a special sensitive region. JGA is a spatial sensitive region formed by cellular modification in the distal convoluted tubules and the afferent arteriole at the location of their contact. A fall in GFR can activate the JG cells to release renin, which can stimulate the glomerular blood flow and thereby GFR back to normal. A comparison of the volume of the filtrate formed per day, 180 liter per day, with that of the urine release 1.5 liters suggests that nearly 99% of the filtrate has to be reabsorbed by the renal tubules. This process is called reabsorption. The tubular epithelial cells in the different process is called दोस्तों हम पढ़ने जा रहे हैं फिर से tubular epithelial cells in different segments of nephron perform this either by active or passive mechanism. For example, substances like glucose, amino acid, sodium, etc. in the filtrate are reabsorbed actively, whereas the nitrogenous waste are absorbed by passive transport. Reabsorption of water also occur passively in the initial segments of the nephrons. During urine formation, the tubular cells secrete substances like H plus, K plus, and ammonia into the filtrate. Tubular secretion is also an important step in urine formation as it helps in maintenance of ionic and acid-base balance of body fluid. Function of the tubules और इसमें हम पढ़ेंगे PCT proximal convoluted tubules. PCT is lined by simple cuboidal brush bordered epithelium which increase the surface area for reabsorption. Nearly all the essential nutrients and 70 to 80% of electrolytes and waters are reabsorbed by this segment. PCT also help to maintain the pH and ionic balance of the body fluids by selective secretion of hydrogen ions, ammonia and potassium ions into the filtrate and by absorption of SO3 minus from it. Henley's loop. Reabsorption is minimum in its ascending limb. However, this region plays a significant role in the maintenance of high osmolality of medullary interstitial fluid. The descending limb of loop of Henle is permeable to water but almost impermeable to electrolytes. This concentrates the filtrate as it moves down. The ascending limb is impermeable to water but allows transport of electrolytes actively or passively. Therefore, as the concentrated filtrate passes upward, it gets diluted due to passes of electrolytes to the medullary fluids. DCT, distal convoluted tubules. Conditional reabsorption of Na plus and water take place in this segment. DCT is also capable of reabsorption of SU3 minus and selective secretion of hydrogen and potassium ions and NH3 to maintain the pH and sodium potassium balance in blood. और अब हम पढ़ेंगे collecting duct. This long duct extends from the cortex of the kidney to the inner part of the medulla. Large amount of water could be reabsorbed from this region to produce a concentrated urine. This segment allow passage of a small amount of urea into the medullary interstitium to keep the osmolality. It also play a role in the maintenance of pH and ionic balance of blood by the selective secretion of H plus and K plus ions. Point number 19.4, Mechanism of concentration of the filtration. Filtrate. Mammals have the ability to produce a concentrated urine. The Henle's loop and vasa recta play a significant role in this. The flow of filtrate in the two limbs of Henle's loop is in opposite direction and thus form a counter current. The flow of blood through the two limbs of vasa recta is also a counter current pattern. The proximity between Henle's loop and vasa recta as well as counter current in them help in maintaining and increasing osmolarity toward the inner medullary interstitium. Example for 300 mole or most mole per liter in cortex to about 1200 mole per liter in the inner medulla. This gradient is mainly caused by NaCl and urea. NaCl is transported by ascending limb of Henle's loop which is exchanged with the descending limb of vasa recta. NaCl is returned to the interstitium by ascending portion of vasa recta. Similarly, a small amount of urea enters the thin segment of the ascending limb of Henle's loop, which is transported back to the interstitium by collecting tubule. The above described transport of substances facilitated by the special arrangement of Henle's loop and vasa recta is called the counter current mechanism. This mechanism helps to maintain concentration gradient. In the medullary interstitium, presence of such interstitial gradients helps in an easy passage of water from the collecting tubules, thereby concentrating the filtrate. Human kidney can produce urine nearly 4 times concentrated than initial filtrate form. Regulation of kidney function. The functioning of the kidney is efficiently monitored and regulated by hormonal feedback mechanisms involving the hypothalamus, JGA, juxtaglomerular apparatus, and to a certain extent the heart. Osmoreceptors in the body are activated by changes in blood volume, body fluid volume, and ionic concentration. An excessive loss of fluid from the body can activate these receptors, which stimulates the hypothalamus to release antidiuretic hormone or vasopressin from the neurohypophysis. ADH facilitate water reabsorption from later part of the tubules, thereby preventing diuresis. An increase in the body fluid volume can switch off the osmoreceptors and suppress the ADH release to complete the feedback. ADH can also affect the kidney function by its constrictive effect on blood vessels. This causes an increase in blood pressure. An increase in blood pressure can increase the glomerular blood flow and thereby the GFR. JGA play a complex regulatory role. A fall in glomerular blood flow, glomerular blood pressure, GFR can activate the JG cells to release renin, which converts angiotensin in blood to angiotensin 1 and further to angiotensin 2. Angiotensin 2 being a powerful vasoconstrictor increase the glomerular blood pressure and thereby GFR. Angiotensin 2 also activates the adrenal cortex to release aldosterone. Aldosterone causes reabsorption of Na plus and water from the distal part of the tubules. This also leads to an increase in blood pressure and GFR. This complex mechanism is generally known as the renin-angiotensin mechanism. An increase in blood flow to the atria of the heart can cause the release of atrial natriuretic factor. ANF can cause vasodilation, dilation of blood vessels, and thereby decrease the blood pressure. ANF mechanism therefore act as check on the renin-angiotensin mechanism. Micturition. Urine formed by nephrons is ultimately carried to the urinary bladder where it is stored till a voluntary signal is given by the central nervous system CNS. This signal is initiated by stretching of the urinary bladders as it gets filled with urine. In response, the stretch receptors on the walls of the bladder send signals to the CNS. The CNS passes on motor messages to initiate the concentration of smooth muscles of the bladder and simultaneous relaxation of the urethral sphincter causing the release of urine. The process of release of urine is called micturition and neural mechanism causing it is called micturition reflex. An adult human excretes on an average 1 to 1.5 liters of urine per day. The urine formed is a light yellow colored watery fluid which is slightly acidic pH 6 and has a characteristic odor. On an average 25 to 30 gram of urea is excreted out per day. Various conditions can affect the characteristic of urine. Analysis of urine helps in clinical diagnosis of many metabolic disorders as well as malfunctioning of the kidney. For example, presence of glucose, glucosuria, and ketone bodies, ketonuria in urine are indicative of diabetes mellitus. And now role of other organs in excretion. Other than kidneys, lungs, liver, and skin also help in elimination of excretory waste. Our lungs remove large amount of carbon dioxide, approximately 1200 ml per minute, and also significant quantities of water every day. Liver, the largest gland in our body, secrete bile containing substances like bilirubin, biliverdin, cholesterol, degraded steroid hormones, vitamins, and drugs. Most of these substances ultimately pass out along with digestive waste. The sweat and sebaceous gland in the skin can eliminate certain substances through their secretion. Sweat produced by the sweat gland is a watery fluid containing NaCl, a small amount of urea, lactic acid, etc. So the primary functions of sweat is to facilitate a cooling effect on the body surface. It also help in removal of some of the waste mentioned above. Sebaceous gland eliminate certain substances like sterols, hydrocarbons, and waxes or sebum. This secretion provides a protective oily covering for the skin. Do you know that a small amount of nitrogenous waste could be eliminated through saliva to disorder of the excretory system? Now we will be talking about disorder of the excretory system. Malfunctioning of the kidney can lead to accumulation of urea in blood, a condition called uremia, which is highly harmful and may lead to kidney failure. In such patients, urea can be removed by a process called hemodialysis. During the process of hemodialysis, the blood drained from a convenient artery is pumped into dialyzing units called artificial kidney. Blood drained from a convenient artery is pumped into a dialyzing unit after adding an anticoagulant like heparin. The unit contains a coiled cellophane tube surrounded by a fluid having the same composition as that of plasma except the nitrogenous phase. The porous cellophane membrane of the tube allow the passage of molecules based on concentration gradient, and nitrogenous waste are absent in the dialyzing fluid. These substances freely move out, thereby clearing the blood. The cleared blood is pumped back to the body through a vein after adding antiheparin to it. This method is boon for thousands of uremic patients all over the world. Kidney transplantation is the ultimate method in the correction of acute renal failure. A functioning kidney is used in transplantation from a donor, preferably a close relative, to minimize its chance of rejection by the immune system of the host. Modern clinical procedures have increased the success rate of such a complicated technique. Renal calculi, stone or insoluble mass of crystallized salt, oxalates, etc. formed within the kidney. Glomerulonephritis, inflammation of glomeruli of kidney are known as glomerulonitis. Thanks for watching friends. बहुत बहुत धन्यवाद.